So welcome back again. This is Empathy Live Lessons, and tonight we're going through Biology Paper 2. So this is going to be after the half-term break. I'm checking, and as you can see, Biology Paper 2 is on the Wednesday, the 15th of June, so quite late on. Now, there isn't much to go through tonight because the advanced information, well, there isn't much. They've eliminated lots of stuff for some reason, but likewise, I urge you to please look at the advanced information and just do everything else found in, let's say, revision guides and so on. Just do everything except what is specifically like in the not assessed section. So tonight I've adapted the presentation slightly. So if you see it like a little symbol, a bit like this is like the in the not assessed bit. So we start off with the Unit 5, homeostasis and response. And we can already see first that here we start with the nervous system, and it's in triple science, foundation and higher, but not in combined science, not at all. So to start off, we start with the nervous system. So these are a network of nerves and synapses all connected together to receptors and effectors, so that humans can react quickly to the environment around them. You'll be familiar with this little pathway here. So you start with a stimulus, something that changes in the environment. For example, that could be something even basic, such as an object moving towards you. Then your receptors are these nerves that will carry these information or through electrical impulses to the central nervous system, which is either the brain or spinal cord, which sends back more electrical impulses, which go through the effectors and back out through your response, basically in response to the stimulus. For example, if you touch a hot object, after this information is sent round in almost an instant, you'll respond by pulling your hand out because it's hot. So we need to learn about the structure of the nervous system. So first we need to know about the synapses. So these are like where two neurons meet, like a junction perhaps. So let's say when an electrical impulse reaches the end of a first neuron, like this here, you've got these electrical impulses coming down. Now what happens is, when it reaches the end of the first neuron here, it releases neurotransmitter molecules. What happens is, these molecules will then diffuse us across a very small gap, which is like nanometers, because diffusion works better at smaller distances, which you should know from biology paper 1. So what happens is that these neurotransmitter molecules will diffuse across and start off another electrical impulse in the other nerve, or the next neuron. So that will pass down information. These synapses are within these things called reflex arcs. So these are along the spinal cord, where it doesn't have to go through up to the brain and back down, even though it's quite quick. So these follow a similar path to the one on the previous slide, so we need to add a few extra parts. This time the sensory neurons will connect to receptors and the CNS, so this will be the spinal cord. And this CNS is, as I've said, the spinal cord, but will also contain a relay neuron. So these will connect a sensory neuron to a motor neuron. And this motor neuron will then go to the effectors. And these synapses connect each neuron. So what you end up having is a stimulus, then your receptor, then your sensory neuron, then the central nervous system or the reflex arc, or the reflex, or the actually the relay neuron, then to the motor neuron, then to the effector, which controls your response. And I'll highlight where synapses are used. So like I said, using this reflex arc, how can we describe how, let's say, someone will pull their hands away immediately when they touch a hot object? So we start with a stimulus, so the stimulus will be the hot object that someone accidentally touches. This then gets detected in like temperature, like receptors in the skin. So that's on your hand. Now this information gets passed on to the sensory neuron. That then travels along the neuron and then reaches a synapse and passes on its information via a a relay neuron. After that, the information gets passed on to a motor neuron, then the effector, and then you respond from your electrical impulses, like the effector and response will be in your muscle, which will contract in a way so that your hands will let go of that hot object and then prevent you from having any further damage to your skin. So that's one example of a reflex arc. And remember, you've just got to identify like possible stimuli, the stimuluses, and the responses after. And just basically in between, it's pretty much the same, except for the receptors, really. Because, for example, light is detected with the um, like a receptor inside the eye, which is yeah, the optic nerve. And for example, in the ear to hear sound, that will be the auditory nerve. These will connect to the neurons again, the relay neurons, then to the motor neurons, then to the effectors, which are usually muscles. Now, next, for like triples again, triple science only, it's another required practical, and this is involving reaction times. So you'll often investigate the effect of a condition, let's say having a stimulant like coffee, on, let's say, how would it affect someone's reaction time. Now, to measure the reaction time of someone, you could use, for example, a computer or device, such as reacting to a change in the screen, let's say um, change in color, and then you press a button when that happens. So would be your reaction time. However, if you don't have access to this in this practical, you could use a falling ruler. So that's where one person will drop the ruler and the other has to catch it. Now, to make things fair and valid, the person catching the ruler should have their hand still and open until they see the ruler drop. Then, the person dropping the ruler should not push the ruler down quickly but they should still just simply drop it without any warning. And just before they drop the ruler, the 0 centimeter end of the ruler should be near the catcher's fingers. Then lastly, when you record these results, you should record them from the top of the finger. So let's say you'll have your finger here. And then you'll have your ruler. You have to measure from roughly here. And then you record the average distance, and remember that longer distances will indicate longer reaction times. Then, after you've done a few trials of this, you should do the whole experiment again. So you do it first as a control, like with normal conditions, like your normal reaction time without any influence of, let's say, any stimulants like coffee. And then you do the experiment again after you've taken the coffee, for example, and you've got to wait for a bit, let's say 10 or 15 minutes, because these substances take time to have an effect. And remember, don't forget to control other variables, such as the hands that someone uses to catch with. So if they're going to catch with their left hand, they must do it with the same hands all the time. Next for triple science is another use of the nervous system, which is thermoregulation. So this basically controls how like your temperature will vary in response to your own temperature or the surroundings. For example, if you're too cold or you're losing heat easily, your temperature receptors in the skin will detect this, or also in your brain, the hypothalamus. Remember, these even the receptors in the skin are quite sensitive as well. They can detect very small changes in temperature, even as much as half a degree centigrade. Then, the thermoregulatory center coordinates a response to increase the blood temperature. So first, the blood vessels near the skin will close and let less blood flow near the skin to lose less heat. That's because if you remember, the blood is hot because it passes by the cells which are carrying out constantly aerobic respiration. That releases energy and makes the blood hot. And what you don't want is the hot blood or warmer blood flowing near the edge of your skin, because that puts it closer to the colder surroundings. That will lose more heat. So that's why you will look more pale, because less blood is flowing through, and your blood vessels will tighten up. That's called vasoconstriction. Next, the hairs will lie vertically up and rigid, which reduce the surface area where heat can be lost. And that also makes a little layer of insulating air. So if I draw the skin slightly, what you'll have is with these hairs, they'll be held up slightly by a bit of skin. And what this will do, it will trap a small layer of air, which acts basically like a coat, almost like this. Because if you've got like an insulating layer, there's going to be less temperature difference between you and your surroundings. And lastly, the skeletal muscles will contract more, and this means it will carry out aerobic respiration more to get more energy to contract the muscles. Yeah, and that means it uses respiration to release more heat. So that's, in other words, it's shivering. Now, the opposite side, what happens if your body gets too hot? Again, the temperature receptors in the skin and brain will detect the high temperature, and again, the thermoregulatory center coordinates a response to decrease the temperature this time. So the first thing is is pretty much the opposite. This time, the blood vessels near the skin open, so lets more blood flow to the surface of the skin or near the top of the skin. And that means it gets more exposed to the slightly colder surroundings, which will try to release more heat. It's basically the blood vessels near the skin open or loosen more, called like vasodilation. So that's why you'll often see yourself get flushed, red, perhaps. Next is the most obvious for us. Sweat is released from the glands. So this uses a thermal energy from our body to turn this liquid into a gas. And that's also a link to physics as well, with the specific latent heat of vaporization turning the liquid to a gas, because that requires energy. And lastly, the hairs lie flat on the skin, and this will increase the surface area for heat to be lost. Now we're moving on to something that's in all groups and tiers, which is hormonal control. So, if you didn't know already, hormones are chemicals which are released by various glands mainly the pituitary glands. That's called the master gland because it releases many hormones. These will target a specific organ which carries out some action. These hormones are carried in the blood and will take long to affect. In other words, it will have to travel around the blood until it reaches a high enough level and it's detected by the organ you want. However, their effects are much more long-lasting. One example could be controlling glucose. For example, if your glucose level is too high, the pancreas will detect this and it will release insulin. Next, the insulin causes glucose to enter the liver and muscle cells, which is stored as glycogen, which is larger and insoluble, so it won't move anywhere. So that means your glucose level is reduced back to normal levels. Now for higher tier, on combined and triple, you'll be asked what happens as well if the glucose level is too low. So first, the pancreas will detect this, and this releases glucagon. This will do the opposite, and this causes glycogen stored in the liver and muscles to break back down into glucose. And that means glucose will diffuse back into the blood, and that means the glucose levels will rise to normal levels once again. However, some people can't control their glucose levels, and this is called diabetes. There are two types of diabetes. We'll start with type 1 first. This is where little to no insulin is produced, and this disease is often inherited genetically. This is from parents, for example. To combat type 1 diabetes, they will often need to control the diet and consume less sugars and carbohydrates. So this means the glucose level after you eat, like when the glucose will diffuse into the blood at the small intestine, as you know, from digestion, the glucose level won't just rise as much. So you won't need, for example, to exercise as much, but it's still recommended. to use the excess glucose in aerobic respiration for exercise, which reduces it further. Another thing type 1 diabetes people can use are insulin injections. So basically, remember, they don't produce insulin, but it can still work on them. However, with type 2, they still produce insulin, but their body does not respond. As in, for example, the glucose does not convert into glycogen and it won't be stored in muscles or the liver. Now, this can be inherited, as in passed on genetically, or you could have like this develops from other diseases. For example, obesity. Obesity will increase your risk of having type 2 diabetes. Now, as a response, they are similar to type 1 diabetes, which is having a controlled diet, which will be less sugar and carbohydrate consumption and more exercise. Another type of hormone some people may need to know about is the hormones in the menstrual cycle. So this will not be on the triple higher paper or the combined foundation paper. So that means it's likely going to be on the combined higher tier and the triple foundation tier paper. So you'll need to be familiar with all four of the hormones in the menstrual cycle and how they all link together. So the first, at the start of the cycle, FSH is released from the pituitary gland. When it reaches the ovaries, it will cause an egg to mature, and this stimulates or causes the ovaries to produce estrogen. Now, this hormone, estrogen, is released and it will stop the release of FSH, so it stops that first hormone from being released, and this causes the pituitary gland to produce LH. This also causes the uterus lining to grow. Now, LH is produced by the pituitary gland when that estrogen is released, which causes the ovaries to release an egg. That's ovulation. Now, after the egg is released, the remains of the follicle, so if I draw a small diagram again, so you'll have let's say your ovaries here. This is your fallopian tube. So when an egg is released, it's from its little shell here. It gets released here, and this egg travels down. But all these remains here, which is called the follicle. Which is highlighted in yellow. That's what causes progesterone to be produced. So this will stop the two other hormones from being produced, FSH and LH. Now, this progesterone will maintain the lining of the uterus until around 14 days more. So that will maintain the lining of the uterus. If, for example, the egg gets fertilized by a sperm cell, and then it allows it for an embryo to develop, when that fertilized egg will implant itself into the uterus. Now, if the egg does not get fertilized, what happens is that the progesterone hormone will fall again, and that will cause the uterus lining to break down, and that's when you have your period or menstruation. Some more hormones are for triple science only. An example can be plant hormones. Now, the most common example would involve auxins, along with the roots and shoots of a plant. So these auxins are a type of hormone that are produced in the tip of the root or shoot, but will move depending on the condition. Auxins will also change how quick cells can divide or grow. This also depends if it's in the roots or shoots of a plant. So these auxins will cause something called tropisms. The first you need to know about is phototropism. So this is where the shoot should grow to a light source, for example, the sun, or a lamp, for example. Now, how does this happen? First, if I draw a shoot, like this, then we'll put our light source right here. So that's our shoot. What happens is, first, auxins are produced at the tip of the root, of the shoots actually, which is right here roughly. However, they get pushed towards the shadier side of the shoot. So they'll concentrate. If I draw auxins in red, they'll start to dot around more on this side because it's darker. Now, this causes an uneven distribution of auxin. Now, in the shoot, auxins will promote cell growth. It will make cells divide faster or grow faster. So that means the shady or dark side of the shoot will grow faster. So if we imagine them like arrows for growth, this side grows much faster than on the left. So that means eventually this shoot will start to almost bend in an exaggerated way towards the light source, where until then you'll have an even distribution of auxin, and then the shoots will just go closer and closer and closer to the light source. Now, the last thing you'll need to know is that this causes the shoot to bend towards light, and this is called positive phototropism. And remember that photo is to do with light. The other type of tropism you need to know is geotropism. So this is to do with gravity. So this will cause roots to grow towards gravity and cause shoots to grow up away from gravity. Now, once again, auxins are produced at the tip of the roots and shoots, but this time they're concentrated on the lower side, pulled by gravity. So if I draw a shoot once again, this time at the side, and I also draw, let's say, a root, which is a bit more wiggly like this. If I like little dot to place little dots where the auxins are, they will be on the lower side towards gravity. So these auxins get pulled down. Once again, this causes an uneven distribution of auxin. Now, this time, auxins promote cell growth in the shoot, like what we said before, but inhibit or slow down cell growth in the root. So if I draw cell growth with blue arrows this time, in the root, the bottom side will grow a lot slower, whereas the top will grow a lot faster. So overall, this causes the root to bend downwards, whereas in the shoot, we know that auxins promote cell growth. They'll speed up cell growth. So this side will grow faster, the bottom side, than the top. So overall, this causes the shoot to first bend upwards. Lastly, you'll need to know about the positive and negative, like geotropism. So if it grows towards gravity, it's called positive geotropism. That happens in the roots, where if it bends away from gravity, the shoots, that is negative geotropism. It will act against gravity. And once again, for triple science, there is a required practical. This is investigating the tropisms. So here's a general method for either tropism. So first you'll grow some seedlings, for example, mustard seedlings, in two petri dishes. And you'll put, let's say, 10 in each, and then you'll line each of these petri dishes with some moist cotton. So let's say with a bit of water in the same volume for each. So you'll put some water on the cotton, place 10 seeds on each, or seedlings, and then you should water the seedling with a fixed amount of water or volume of water every day, and do this for 3 days so that they start to grow slightly. Then you discard all the seedlings that have not germinated, as in they have not sprouted up. So they'll look more. So here's your seedling. If you start to see a little green shoot coming out like this, that has germinated. Whereas if it just stays as it is, like this, just a seedling, unfortunately it hasn't germinated. So what you do is you discard all the seedlings that are not germinated, and then you equalize the number of seedlings in each dish. So you basically match the number of seedlings in each dish. So what you don't want is, let's say, 6 seedlings to be in one dish and 8 to be in another. They need to have the same number, and you can't transfer one seedling to another. So once you've equalized the number of seedlings, you'll place one dish in the condition you want to investigate. That could be light, gravity, or the direction of light. For example, light you could just leave by windows, like top. To investigate the direction of light, you could put it in a box, cut out a hole at the side, and shine some light into it. And if you want to investigate gravity, you could put the petri dish on its side, or just leave it as it is. And then you place the other petri dish in an area where the condition has no effect as a control. For example, if you are investigating light, you'll put one petri dish in light and the other petri dish not in light, so light has no effect. If you're investigating gravity, your control would be putting the petri dish onto like a motor that's sideways, like this. So you'll have a motor. You'll have like a little shaft, perhaps. This will hold your petri dish. And this will keep rotating it around. This will basically have have like gravity of no effect because gravity constantly acts in different directions. Now to investigate the effects itself, you'll need to leave the petri dishes of seedlings in each condition for 4 days. So leave one in light, for example, and another one not in light. You'll do that for 4 days at the same time, and you'll water each petri dish again every day with a set volume of water, and remember to measure the length of the shoots or roots with a flexible ruler or string, because usually what you'll have is, if I draw the seedling, you'll have the shoot coming out, and it will bend to the side often. So what you could do, perhaps, is get some string, then measure it, or you could just use a flexible ruler. The next unit we're going to move on to now is genetics and evolution. So first is some triple science content, or that's only found in the triple science paper, foundation and higher. This is meiosis. Now from paper 1, you should know about the other cell division process of mitosis. So meiosis is also similar, involving cell division, but it's quite different. So first you start with one cell of 46 chromosomes. So we're talking about human cells here. Next, the DNA and genetic material duplicates inside the cell. Next, the cell divides to form two cells with 46 chromosomes each. Then, each cell divides again to form four daughter cells with 23 chromosomes each. So this is what it looks like. So you have one cell with 46. This becomes a larger one with 92. Then it will split up into two daughter cells with 46 chromosomes each. Then it'll split up again to form four daughter cells with 23 chromosomes each. So these daughter cells, however, will be genetically different from each other. So meiosis is used for producing sex cells or gametes like sperm and egg cells. So when the two gametes fuse, their genetic material is combined together to make a full set of 46 chromosomes, or 23 pairs. So this is sexual reproduction. That means you'll get one set of chromosomes from your father and another set of chromosomes from your mother to get 23 pairs of chromosomes. On the other hand, you'll need to know about asexual reproduction. So this can be done with a single parent. So what essentially happens is they clone themselves as their genetic material is not mixed with others. So this type of reproduction only happens with mitosis, as it produces daughter cells with the same genetic material to the parent and each other. Next in genetics is the genome and DNA. So this is on everyone's paper except combined higher tier. So the DNA contains the genetic material of many organisms in a double helix structure from two strands. So you'll be familiar with its shape like this. Now, these strands will contain bases, so little almost ladder-like things. And what these bases do, they're little chemicals that attach to the strands. And these will be read in order, like instructions, and then will code for specific amino acids. And these amino acids will make specific proteins with specific structures or shapes. One example could be enzymes and their active sites. Now, a gene is a section of DNA that will code for some proteins, and that will make a characteristic, such as eye color, hair color, even height perhaps. Now, the genome is the entire code of DNA for a single organism. So that'll be all the chromosomes, for example, in the human. Now, for humans, we understand the genome and it's quite useful in three ways. First, we can learn our migration patterns. For example, we can see where many humans would have mixed with others and perhaps bred and then produced offspring where the DNA mixed together. So we can trace back. The second way is we could look at how diseases or disorders can be inherited. So we're going to look at a few inherited disorders in a bit, such as polydactyly, cystic fibrosis. Those are the two which you need to know. You also need to understand that the genome was also useful for understanding how some genes can cause diseases, like diabetes, which could be inherited genetically. Now, Triple Science higher tier need to go further into the structure of DNA. They need to know more detail. This is a common exam question. It often carries 4 marks. So describe the structure of DNA. You'd first get it by saying it's a polymer, or a chain of repeating units in a double helix shape. So that's like a twisted ladder almost. Now, each monomer, each unit, is made up of a sugar and phosphate. So that forms the backbone. So this is your backbone here. And then bases are the ladders which you have, the little steps. If you draw a single monomer, you wouldn't need to know how they exactly bond, but to draw a single monomer of DNA would be starting with the simple sugar, which is often drawn as a pentagon, like this. Then that bonds to a phosphate. In this rectangle bit, there's a base. So this entire bit here this entire unit here is the monomer or the repeating unit of DNA. Now the third mark would be to mention that there are 4 different bases, A, C, T, and G. You don't need to know the actual names, but that's all you need to know. And lastly, you'll need to know that since you've got a double helix or 2 strands, so that the backbone kind of is on mirrored on the other side, the bases are also like paired up, if you like. So if we draw out 2 bones like this, we'll have a little bond here, and we'll have some bases. For example, we'll focus on 2 bases here. Now, bases from each strand are paired complementary, so A on one side will be paired with T on the other side, and G on one side will be paired with C on the other side. And one way to remember this, A, T, G, and C, is all thieves get caught. Now, furthermore, triple higher tier will also need to know about mutations. So remember that mutations, which will come into, will change the DNA structure or the sequence, but the sequence is already important. So as you knew that the new protein structures are made, they're made up of the DNA sequence being read in the ribosomes. That's protein synthesis. So 3 bases will code for a specific amino acid. So every 3 bases, an amino acid is fetched, and then it starts to build one big structure, the protein. Now, many of these amino acids then form the protein, which I said, and then this will cause the protein to function in a certain way, such as for strength, structure, maybe to catalyze reactions, such as enzymes for digestion. However, not all of the DNA sequence codes for amino acids, so they can turn genes on or off. So if they, for example, you don't want the gene for eye color to be on or expressed when you're, like, let's say, a cell in the skin. You don't want your skin turning a different color, weirdly. And this is called a non-coding DNA, so make sure that only specific parts perhaps are read or expressed. Now, mutations are changes to the DNA sequence. So if you remember, the DNA is read in the ribosome, and three bases are read, and they will cause a specific amino acid to be fetched by what's called a carrier molecule. Now, when DNA is copied, such as in mitosis, there will be mistakes. It will be hard to perfectly copy DNA all the time, so mutations happen all the time. Now, a small number of mutations can change the shape of a protein because they can change occasionally which amino acid is fetched. So they can change the shape of the protein very slightly, but it won't affect its function completely, such as an enzyme's active site. So let's say this is an enzyme. So with some mutations, it would look slightly different, perhaps something like this. It would still work though. However, if we've got so many mutations, so many changes to the DNA sequence, very different amino acids would be fetched because when the DNA is read in pairs of three bases at a time, different amino acids are fetched, and that means this builds a different protein, and it could look something quite different, such as this. So that means, for example, the active site has changed now. Let's say with denatured enzymes already. So that means, for example, here the active site no longer fits to the substrate, which is a link to paper 1, and that means you can't catalyze some reactions anymore. Now, moving back to some content covered in all papers except combined higher tier is genetic inheritance. So a recap from sexual reproduction is that each parent contributes half of their own DNA, so they'll have 23 pairs of chromosomes, or perhaps pairs of genes. So as I've said, genes control characteristics. They'll code for some DNA, they'll make a protein which controls a characteristic. Now, one of these genes from the pairs is contributed to the next child to inherit. So a gene contributed is called an allele, which is a form of a gene. Now this might ring some memory bells. These alleles can be either recessive or dominant. Now, to show genetic inheritance, we need to show alleles. So when each allele is inherited from two parents, they may interact to express or to show a characteristic. Now, to show a characteristic, or the appearance, that is called the phenotype. Now, for example, if one dominant allele is present in the pair, the dominant characteristic is expressed, and you'll be told about this in the question. So you'll be told what the dominant characteristic is. For example, it could be one type of hair color. Likewise, you'll be told about the recessive characteristic, but only recessive characteristics are expressed or shown when there are two recessive alleles. So if I show, let's say, a dominant as a D, recessive as an R, you have three combinations really. So both are dominant. That means there's a dominant characteristic. If you have one dominant, one recessive, that's also a dominant characteristic. If you have two recessives, as in nothing that's dominant, the other recessive characteristic is expressed. Now, we can show some inheritance again by showing the alleles. And we use something called a Punnett square often. It's a funny name, but you'll recognize it soon. So remember, dominant alleles are represented with capital letters, and recessive alleles are written as lowercase letters. You'll also be given the following. So each parent, remember, they can contribute one of their alleles to their offspring as they're found in pairs. So each allele can sort of match up or pair in 4 different ways or 4 different combinations. And you'll need to know the following terms: heterozygous, so the two alleles are not the same. And lastly, homozygous, where the two alleles are the same. So we'll have an example here. So let's say the allele for normal health is represented with a capital F, and the allele for cystic fibrosis is represented with a lowercase f. So this tells you that cystic fibrosis, which is a condition inherited genetically and it's to do with the lungs and makes breathing slightly harder, this is a recessive characteristic because it's controlled by a recessive allele. So let's say two parents who are carriers of cystic fibrosis. Now this is a new term. Like they have this carriers mean they have normal health, but contain or can pass on the condition. So the parents are carriers, so they have normal health, but they contain the allele for cystic fibrosis, and they can potentially pass on the condition. So let's say they have a baby, and we want to calculate in theory, the probability that the baby will have cystic fibrosis. So both parents will have a genotype for the pairs. So both parents will have normal health, so they'll have at least one dominant allele. However, they are carriers of cystic fibrosis, so that means they will have an allele for cystic fibrosis. So their genotype, their pairing of alleles, is capital F, lowercase f, like this. Now, to find the probability, we'll first draw a little Punnett square, which looks like a multiplication grid. So you'll have one parent on the top row and one parent down the side. And what you'll put is each of their alleles. So let's say parent, there's the same anyway. We'll put parent 1 at the top and the parent 2 at the side. So parent 1 will have capital F and then a cystic fibrosis allele, the lowercase f. And then parent 2 will have something similar, capital F, lowercase f. Now, what we do is, for example, we'll pair each of them up. So we'll draw a line down here, and where they intersect, that's where we'll draw the new combination. So this capital F will combine with this capital F. This capital F will combine with this one here, and they'll make the combination capital F, lowercase f. This will make this. And again, you'll finally get this. Now, cystic fibrosis is a recessive characteristic. That means you need both recessive alleles for someone to have cystic fibrosis. So again, cystic fibrosis, it's recessive. So you need 2 or recessive alleles. So that means you need the pairing f, lowercase f, lowercase f. Now we can find that here. So we need to circle that. And we can see there are a total of 4 combinations. So that means we've got one combination out of 4. So that's a 1 in 4 chance, or 25%. Now, as a bit of information before we move on, you'll need to know about the other inherited disorder, which is polydactyly. Now, you'll need to know that polydactyly is caused by a dominant allele. You could then be asked on something similar like this, but with polydactyly, and you'll just need to sort of figure it out for yourself. Now, moving on into our last unit now is ecology. So first, we want to know a few definitions. This is mainly for combined foundation, which is the first part. Now, in biology, what's meant by, especially in an ecosystem, by a population, a community, and some factors like biotic and abiotic ones. Now, a population is just the number of organisms. That, for example, could be the number of fish in a pond. Now, a community would be the total of all populations. Well, what happens is these populations or these different organisms will then interact with each other in different ways. So that's actually the biotic factors. So living factors that can affect or interact in an ecosystem. Whereas abiotic factors are non-living factors instead. Next, in this little section is to do with the interaction of organisms. So this first we'll look at interdependence. Now by the name, what does this suggest about many different organisms in an ecosystem? So dependence means, well, you depend on someone, you rely on someone, you need them to survive. So some organisms depend on others. Now, in what ways could organisms be dependent on each other? One way could perhaps be for food or shelter. Another could be competition, as in they could kill other organisms that would be harmful to you as well. Now, one good example could be bees and flowers. These are two organisms that heavily depend on each other. This is because the bees will do what the flowers want. So these will spread the pollen for them. Whereas the flowers will do something for the bees. These will offer nectar in return. Now, another way some organisms could interact, however, is not very helpful to each other. This is competition. So organisms will have to compete for things that help them survive. Now, for example, they could be, for animals, let's say food, mates, as in a partner to breed with, and produce offspring, or even territory or space, or just somewhere to live, perhaps. Now, plants also compete for things that help them survive. That would include things like light, space, well, it could even be water and mineral ions in the soil. That is why you often see not many other organisms like bushes, flowers, and so on near trees, because trees, well, they hard to compete with because they have really large roots which take up loads of space and lots of water as well. And don't forget the trees are big, they will take up lots of light, and not much will get down to other plants. Now, we're going to look at the factors themselves in more depth. So, abiotic factors were the non-living factors of an ecosystem that affects how organisms can live. For example, temperature, rainfall, other weather climate related things. For plants, it could be soil with their minerals and pH, carbon dioxide levels for photosynthesis, and perhaps oxygen levels for animals. This can link in with altitude as well. Whereas, let's say, biotic factors, these are living factors that can affect how organisms can live in an ecosystem. These could be, as said, other organisms through competition, diseases, and availability of food. Now, this links into some levels of organization. So these are what you know as food chains. So what's went by first? Biomass. So biomass is just mass of, let's say, how would I put it? Like animals or organisms. Now, it's better to measure biomass than the population because for producers, as you know, of biomass, these would tend to be your plants at the start. Yes, you could have perhaps grass, which is a high population, but you could have one single tree. And usually you'd expect a higher population for producers, but instead it's just a higher biomass, as in there's lots of mass in a single tree, which could sustain a whole ecosystem with it. Now, one example of a food chain is on your right here. So at the bottom you have your producers. So these are just mainly plants, algae, and so on. These again then get consumed. So first, consumers. Next to the secondary consumers on top. So these will consume the first consumers. And then on top you'll just have more predators. And remember that prey gets eaten by predators. And this arrow will show the transfer of biomass. Not whoever gets eaten, not whoever gets attacked, not whoever's a prey or predator. It's the transfer of biomass. That's what you'd want to answer in your exam. Now, one thing to note is something quite interesting. These are prey and predator population cycles. So let's say we'll take a look at the graph below, and we need to describe and explain the trends of these cycles. So what you can see is you've got the predators, which are the lynxes, and then so the lynx are the predators. So that's in the grey graph or line, and the hares are the prey. And what you can usually see is that the population or perhaps biomass of like the hares is much larger than the biomass or population of the lynx or the predators. And that's because when biomass is transferred, not, for example, not all of the biomass is transferred. For example, the lynxes will not eat, for example, the bones of hares. That because the bones will count as biomass. And perhaps some of the biomass is lost in other ways, such as energy transferred to the surroundings, such as through heat, movements, and so on. Anyways, what we usually see is that the two populations will increase and decrease together. One good example to look at could be this little section here. We'll just take a look at one cycle. So let's say we'll label this point number 1 right here. So as you can see at this moment of time, you can see there are a higher number of hares, lots of hares, and not as many lynxes. And that means there's more prey per predator, it's easier to find hares, or there's less competition perhaps, and it gets harder and harder for the hares to survive because they like they're food to the lynxes. So basically the lynxes, the predators, will have more food available. That means they'll be able to survive much easier and perhaps reproduce. That's why eventually, since there's right, so there's lots of food for the predators. They'll eat more, survive longer, and reproduce. So that means the population of hares increases. Like the hares will decrease since they're being eaten, and the population of lynxes will increase. That's at the next line, just roughly here. Now, that means that there won't be as much like prey or hares per for like all the lynx as like an increasing population. So that means there's more competition for food among the lynxes. And that means these lynxes are less likely to survive. Now, that means as well the population of hares is still decreasing, while the population of lynxes are still decreasing. And lastly, you can see that the population of hares goes back up again. Now, why is that? So that's this little section here. Now, that's because with fewer predators, the hares are more likely to survive and reproduce. So that means their hare population will this time go up. And it restarts the whole cycle once again. The hare population goes up again, while there aren't that many predators, and that brings it back us to 1. And the cycle repeats. Now, for all groups and tiers this time, there is a practical. And you've been taught this likely for weeks ago, probably, in response to this advanced information. So all groups and tiers, so all papers, will have like a required practical on sampling the population or the distribution. So this is one of the practicals you could be asked on. So for example, this could be estimating the sample of a population in a given area, such as the population of daisies in a big field, like a school field. Now, you'll be given some equipment like this in a list, like some measuring tape and some quadrats. So these are little squares like you lay on the ground. Now, these are usually half meter by half meter. You could get slightly larger ones, 1 meter by 1 meter quadrats. And often with these quadrats, you will have little smaller lines like this. These divide up the quadrat even further. So you can, for example, calculate percentage cover. Now for the method itself, first you'll measure the length and width of the place you're sampling, let's say the field. Then you'll use a random number generator to generate coordinates based off the length and width of the field. And I'll draw a little diagram at the bottom. And you'll use these coordinates to produce like a place where you put the quadrat down. So if I have the field here, you could, for example, go along randomly. How much? Let's say 10 meters. Then inwards slightly, some like distance, let's say 2 meters. And then that's where you'll place your quadrat. And then for step 3, you'll count the number of organisms present in it, like the number of daisies. And then you'll repeat steps 2 to 3 again. So you'll get like a random number generator, and you'll find a different place randomly to place a quadrat down. And then you'll sample how many like organisms there are in it. So you'll keep repeating it until you get 10 readings from 10 quadrats. Now, you'll then calculate the mean number of organisms in each quadrat, and you'll use this little formula to calculate the population estimate. So your population is the mean organisms per quadrat divided by the area of all the quadrats multiplied by the sampled area. Another way you could think of is the mean number of organisms per quadrat multiplied by the number of quadrats needed to basically sample or to fill the surveyed area. Now, for example, let's say you had a field that was, let's say, 50 meters long and then 15 meters wide. That would mean that its area is 750 square meters. Now, let's say you used quadrats that were, let's say, 0.5 meters by 0.5 meters. That means they're 0.25 square meters. So the number of quadrats needed to, let's say, fill like the field would be calculated by your area of the field, 750, divided by 0.25, the area of the quadrat, and that gets you 3,000 quadrats. And basically, this is like would be your magic number to multiply the mean, like the number of organisms per quadrat. So you get an estimate for the whole population of, let's say, daisies in a field. Now, the practical could change slightly, and they could ask you instead to investigate the population distribution. So this is another variant of this practical, where you'll instead measure distribution. For example, where are you more likely to find an organism? Are they going to be more likely like further away from something, closer to something? And this is what you'll use. Now, one example could be the distribution of daisies around a tree in a field. Let's say you'll have a tree, and you'll have a big field around here. And you'll expect more daisies to be further away because there's less competition for light, water, soil, and so on. And you expect fewer daisies to be inside or near the tree. Now, one variable that can change should be like only changed. For example, that could be light intensity around the tree. Now, similar equipment is used. For example, the quadrats are used again. This time, transects are used, along with sensors, which are optional. Now, first, with your method, you want to roll out some tape measure, let's say 40 meters long, from the tree, in a straight line outwards. So that would be, so let's say you have your big field again, and your little tree, and you'll want your transect, which is like a big piece of tape, going outwards from the tree, 40 meters. Now first, you'll place a quadrat aligned with 0 meters on the transect. So you'll have your start of your tape, your 0 right here, and you'll place your quadrat first here. Then you'll, for example, record the like number of daisies found, the number of organisms found, or perhaps the percentage cover. So how many squares out of, let's say, the ones on your quadrats are, as in the smaller ones. For example, you could get quadrats with 25 smaller squares, and you'll count how many squares are like half or more covered. So what you'll do is you'll find and record that, and you'll do it again, this time on the other side of the transect, perhaps. So you'll place a quadrant on the other side here, and you'll do the same. Then you'll repeat step 2, so you'll move the quadrant along, let's say 5 meters, moving further away from the tree, and you'll record on both sides of the transect, like for example the number of daisies you found or the percentage cover of, let's say, grass. And then you repeat all this once again with the transect moving outwards in a different direction. And you do this to get 4 sets of data. Don't forget, each time when you record the number of daisies found, you'll record the distance on the transect as well. Some notes are, for example, you may want to measure the variable you're changing, like the light intensity, using its relevant sensor. For example, to measure light, you'd just use a light meter or a light sensor. And remember, do this every time you record the population or percentage cover in each quadrat, or every time you move along your quadrat. Now, figure 2 was, let's say, repeating the experiment. So you'll have your tree here. First, you'll have one transect going 40 meters in this direction. For example, you'll choose another direction going the opposite way in 40 meters. This would be a repeat. Then you'll have another one going another direction. So this you repeat again, and so on. So you'd follow these 3 steps again. And remember to keep moving the quadrat at regular intervals, let's say 5 meters, until you reach the end of your transect. Now, next for this unit, you'll need to know about a few cycles, such as the water cycle, which links into some other subjects. Now, this is on everyone's paper except triple foundation. Now, it's quite simple, really. Water can be transferred from the ground up into the atmosphere. That could be through, let's say, lakes or trees. And that can happen in 2 ways. So if I draw some water here, that can evaporate or just go out the plants themselves by transpiration, which you'll learn in the other paper. Now, water can fall back down to Earth again as precipitation. So that's like your rain, hail, sleet, snow, and so on. And then this returns to the other sources, for example, surface runoff, which is on the surface as said, or per location through groundwater to plants or other water sources like the sea again. Another cycle you'll need to know about is the carbon cycle. So, starting with the strawberry plant, describe how carbon is cycled within an ecosystem. 6 marks. So, we'll start with the strawberry plant. So, with a strawberry plant, carbon is stored mainly in glucose or starch. Now, when the strawberry is eaten by the animals, or us, the glucose is then used in aerobic respiration. Now, as a result, the carbon is now stored within the animal's biomass or as a product of respiration, which is carbon dioxide. Carbon is stored in carbon dioxide. What you could also say is when the animal is consumed again, let's say by predators, the carbon stored in its biomass will transfer to the predator. Now, when any organism dies, that goes for any plant or animal, they are then decomposed by detritivores like worms, and then by microorganisms. So these microorganisms will then feed on this biomass, and then, once again, like us, will use it in aerobic respiration. The carbon is then stored in the products formed, carbon dioxide. So we've got loads of carbon dioxide being emitted from all these organisms. Now, to complete the cycle, this carbon dioxide is then taken in by many plants, like the strawberry plant, and it's used in photosynthesis. Now, along with water and light energy, as you should know, the carbon is then converted into a product, which is glucose, formed from photosynthesis. Now, that's the end of the slideshow. That's the end of this lesson. If there's anything else, please comment down below. I could make some quick videos as well. Otherwise, that's it. Goodbye.